This PowerPoint will cover the gram-negative diplococci and the coccobacilla. Neisseria species are recovered from the human respiratory and urogenital tract, and these organisms are transmitted from person-to-person contact. The normal pathogens, or the most predominant pathogens that are found, are the Neisseria gonorrhoeae and Neisseria meningitidis. These organisms are all aerobic organisms. They are gram-negative diplococci, and they all are oxidase positive and catalase positive. Neisseria gonorrhoeae is the second most commonly reported sexually transmitted infection within the United States, and this causes the infection that we know as gonorrhea. This infection is typically localized to the mucosal surfaces, and with males they usually exhibit uritis with dysuria and urethral discharge, and with females this organism will colonize the endocervix and will cause the vaginal discharge, dysuria, and abdominal pain. And this organism can also infect other sites like the eyes, the throat, the rectum. With these infections, if they are left untreated, then they can disseminate, which can lead to things like the pelvic inflammatory disease or purulent arthritis and septicemia. And because this is transmitted vaginally to the infants, if the mother does have the organism, then the babies are all given a erythromycin optic solution that once the baby's born, they'll put the solution over the eyes, and this helps to prevent any sort of trauma to the infant's eyes from this infection. And if you've ever had a baby or seen any images or pictures of babies right after they're born, you can see where they have that little gel type solution over the infant's eyes. The virulence factor includes the surface villi, which it helps this particular organism to actually transfer genetic material between different strands of the organism. It allows it to attach to the mucosal surfaces, and it also allows it to invade the host. This organism is also able to survive any sort of attack response from the host because it is able to prevent the cells, the neutrophils, from actually phagocytizing this particular bacteria. And for that reason, you can actually see it inside of the white blood cells if you were to do a special stain to look at those. It also has a capsule. It produces an endotoxin, and then the outer cell membrane proteins, they are responsible for eliciting the different inflammatory responses within the host. And then the outer membrane proteins are involved in the attachment and invasion into the actual mucosal surfaces. So when you're working up Neisseria gonorrhoeae, this particular organism will typically not grow on blood agar, but it will grow on chocolate agar. So with this organism, what you're looking for on the chocolate agar is small grayish-white, convex, translucent, shiny type colonies, and they can have smooth edges or slightly irregular edges. This organism is an aerobic organism, so it can grow in the presence of oxygen. And of course it gives a gram-negative diplococci stain when it's gram stain. They are oxidase, catalase positive, and DNase negative. And if the main test that you do to work up these organisms is the carbohydrate utilization test. And so you could do this in a test tube, but here at the bottom there's an image of some more newly developed test methods, and there are several that look similar to this, or they may vary to a certain extent, but inside each of these wells you have the different carbohydrate solutions. And so for this one here, C is your control, and then you have your glucose, maltose, lactose, and sucrose. So you have your sugars that follow. Then you have a DNase test and a beta-lactamase test. But what you're looking for is to see if this organism is able to utilize these sugars. So you're looking for that yellow color change where it has utilized it. And so this particular organism is able to utilize glucose. So your control would be here, which would be a negative. You wouldn't expect any sort of change. If this did change, then your results to follow wouldn't be valid. And so with this particular organism, because it does utilize the glucose, but negative for everything else, so these are all negative, then that gives you your results here, where it's able to utilize glucose but nothing else, and that's how you would differentiate Neisseria gonorrhoeae. Neisseria meningitidis is transmitted through respiratory droplets. It can also be transmitted, it's suspected to be transmitted through sharing of drinks and things like that. It mostly affects the adolescent crowd, and this is usually seen with like college dormitories, military dorms. However, vaccination over the years has helped to control and prevent such a severe form of the disease. It can cause fatal bacterial meningitis in children and adults, and so this is usually accompanied also with a type of rash, a petechia type rash, and it is also associated with bacteremia. And so what this can do is lead to things like thrombocytopenia, DIC, and shock, with the DIC, disseminated intravascular coagulation. So this is where the coagulation is actually affected, and you have clots happening in areas of the body where it shouldn't happen. And then you also have other areas that are bleeding out from where those clotting factors are disrupted. It can also lead to the patient actually bleeding out in death if a patient experiences this. So with this particular organism, it is the leading cause of community-acquired meningitis. Other sites that can be affected would include conjunctivitis, sinusitis, and different infections throughout the organs, and a pneumonia without the actual spinal meningitis, bacterial meningitis. So with this organism, once the patient comes in contact with it, it first, has to use its virulence factors, where it has the surface villi that allow it to attach to the mucosal membranes and actually start invading through those epithelial cells and through the submucosal membrane layers. And then it has the capsule that helps to prevent it from being recognized or phagocytized by the neutrophils. And then once it has survived and is able to grow and spread throughout the bloodstream, then it can start producing a more systemic type infection, and which could also include the release of the endotoxins that can create something like a shock. And then you have the IgA protease that increases the host susceptibility to the invasion, so it actually allows this organism to be a little bit more invasive. And so you can perform different serotyping to help identify this organism, and there are commercial kits out there that help with this. And so when it would be useful is if you're looking for, if you have an outbreak within a college and you're trying to kind of link this outbreak to the same organism, and this is where you would actually do the serotyping. When working up this organism, it can grow on blood agar, but it's not as likely to see it growing on blood agar. So again, this is something that would be cultured on a chocolate agar. So this is where the laboratory has to know the source, so it's very important when submitting samples that the source is reported. Of course, with this particular infection, it would be a spinal fluid, so there would be various tests that would be ran on the spinal fluid, including looking at a gram stain where you're looking for the gram-negative diplococci, and it would be plated on chocolate agar because you would be expecting that this could be a potential cause of the infection if you were expecting meningitis. And so when it grows on chocolate agar, it will be medium to smooth round colonies that are moist, gray to white. And you can have some encapsulated strands, so they may have a mucoid type look to them. And it can give a greenish cast to the agar underneath those colonies. And you can see that a little bit here in this image where it kind of has a tinge to it in that 48-hour growth. These organisms are aerobic, and you can see them intra and extracellular, so you can see them actually inside of the white blood cells. So if you were looking at a gram stain, this isn't a very good image of it, so this would be a cell here, and you can see it looks like it's probably one there. And these are probably also intracellular. You cannot see the outline of that cell, but they do look like they're around the nucleoli or the nucleus here. And so you can see those gram-negative diplococci there. These organisms are oxidase positive, catalase positive, and DNase negative. And again, when you're looking at carbohydrate utilization, these organisms use glucose and maltose. You can see that glucose and maltose there at the bottom have turned yellow. Moraxella catarrhalis. This particular organism is normal microbiota of the upper respiratory tract, and it can occasionally colonize the female genitalia tract. So these organisms are spread because of the patient's normal strand. So it can go from where it would normally be found to a site that would usually be considered a sterile site. And so this is a person-to-person spread through the respiratory droplets. And this particular organism will cause a respiratory tract infection, and it's most commonly localized to sites that are associated with the respiratory tract. So this would be like your ears, sinuses, and it can cause pneumonia. And so a lower respiratory tract infection is often seen more commonly in your elderly patients or those with some sort of like chronic obstruction pulmonary disease, so your COPD. And so with patients elderly patients or COPD patients that present with a pneumonia type infection, then this would be one of those organisms that would be suspected. So this organism grows on chocolate agar with a pinkish brownish opaque type hue to it. These organisms they demonstrate something that's very unique, which they have a hockey puck type a demonstration. So if you would actually use a loop to push this colony, it would actually slide across the agar and not break up. It's not sticky or anything. So that is one of the characteristic things that if a tech is working this up and it looked suspicious to be Moraxella, then they would actually see if they could push that colony across the plate. These organisms are aerobic. They are gram-negative diplococci, oxidase positive, catalase positive, and DNase positive. And these organisms do not utilize any of the carbohydrates, so they would be negative for glucose, maltose, lactose, and sucrose.